Good evening, everyone. Hello, and welcome to Stonehouse in the UK for the third and final session of the Fungi Symposium. My name's John Parker, and I'm Technical Director at the Association. If you managed to join us earlier today, then I hope you're ready for some more amazing presentations as we cover chapters 9 and 10 of Lynne Boddy's new book, Fungi and Trees, Their Complex Relationships. And if you were unable to join us earlier, then don't worry. These are entirely standalone presentations, and watching them now won't spoil the ending of the book for you. When using the chat, please select all panelists and attendees. Feel free to say hello, let us know where you're watching from, but all panelists and attendees, please. Ask your questions using the Q&A panel, stating who your question's for. We won't be able to go through all of them, but everything will be sent through to our speakers afterwards, so they'll get to see all of it. If you haven't bought the books yet, but you want to get involved, then they're available exclusively through our website, and a link, I'm sure, will be in the chat soon. Now, if you're a student, another way to get hold of the books was to enter our Student Fungi Book Competition, where we ask student members to write a 500-word article or record a 3-minute video describing why they decided to join the arboricultural industry or to talk about a subject that interested them. We had loads of entries, and earlier this week, a special panel was convened to select the winners, which I will announce right now. In each category, so that's video and article, the judges selected a third place highly commended award. In the video category, the third place went to Alex Cook, and in the article category, it was Cecily Withall. They will be receiving a book-based prize in due course, so well done to you. In second place for the video category was Simona Gervakate. I've probably just murdered your name there, Simona, I'm sorry, but hey, it's you, Simona, well done. Second place. And in the article category, Lyndon Reed. Congratulations to both. You will also be receiving a bundle of books. And finally, the winners in both categories will be winning an even bigger bundle of books that may well involve books not too dissimilar to this. This was very hotly contested, very close indeed, but in the video category, the winner is Hazel Irving. Well done, Hazel. And in the article category, the winner is Stella Boland. Congratulations, Stella and Hazel. Well done to all of you. Thank you all so much for entering. As a special thank you to all of you, we're also going to do a random prize draw of all of the other entrants, and they'll win something too. So it's prizes for all, pretty much. So that's really good. Thank you very much to the member of the Arboricultural Association who generously donated these prizes, and then also to the second Arb Association member who did the same and donated some additional books to the prize fund. This was a really great gesture. We very much appreciate it. And you'll all be able to see the winning videos and articles soon in the Arb Mag and probably at a forthcoming webinar as well. So congratulations to everyone. Usually at this point, I promote the next webinar in the series, but we're taking a little break after this symposium, for reasons you'll understand if you've been here all day. 3 webinars in one day is too much for anyone. So we'll be taking a little break. We're coming back to webinars on April the 28th, and we'll be promoting that soon, so keep your eyes out for that. But today I get to tell you about something rather different, because tomorrow, April the 1st, sees the start of the all-new qualifying rounds for the Urban Tree World Cup 2021. Every day for 16 days, you'll have the opportunity to vote for your favorite in that day's qualifying group. The 16 group winners will be going into the first round proper to join the last 16 of the last tournament in 2020. Okay, do you get that? So if your favorite tree isn't in one of the groups, don't be upset. They're probably already in the first round by virtue of their performance last year. You can find out more information on our website. The first group is ACER. It's there for you to see, and you'll be able to start voting tomorrow morning. You can vote on social media or on our website. Tomorrow, we'll release the rest of the groups, and we'll put something on the website about the rules. And the first rule will be, this is just a game. Please don't take it too seriously. Lots of people unhappy last year. If you don't like it, go and find another game to play. It's just a bit of fun. The point is just to pick your favorite tree for an urban environment. We know there's loads of variables and complicated things to consider. This is just supposed to be a bit of fun. So behave yourselves. Right, back to the Fungi Symposium. This evening, we'll be hearing from Anne Pringle and Jenni Nordén. But to introduce this session, before we get started, we will turn first, for the final time until the questions, I'm sure there'll be an appearance in the questions too, to Lynne Boddy.
It's a pleasure to introduce you to this evening's session, which is about environmental change and conservation. Now, of course, we all know that our environment is changing. We know that modern forestry practices are very different from the ancient management techniques that we've had for 1,000 years, with coppice and pollarding and wood pasture. We no longer have hardly any remnants even of primeval forest. This was taken in Bielavisia. In primeval forest, it takes you about half an hour to walk, 100 yards, because you're climbing over these huge fallen trunks. There are standing dead trunks. There are large pieces of woody material. And if you compare this with perhaps a managed woodland today, where there will be stumps where trees have been felled, if you're lucky, there might be a pile of logs, and maybe there's logging brash, but perhaps not often very much more. Of course, we do have trees in streets and lining roads. Not only is land use changing, so too is climate, and we're all aware of this. I was lucky enough to be involved in the first published study on the effects of climate change on fungi, on fungal fruiting. And these were revealed by records from the past dating back to 1950 from southern England. And what we looked at was the average date when fruit bodies first appeared in the autumn and the average date when they were no longer there. And that's what's plotted here. So this is time 1950 up till about 2007 in this instance. And this is the day of the year. So this is September-ish around here. This is Christmas time around here. And you can see that, okay, every year weather fluctuates, and this affects when fruiting occurs. But if we can fit a line through this, and the first fruiting date and the average last fruiting date, we'll see that it doesn't actually change very much over the time from about 1950 to about 1978-ish. But after that, the average first fruiting date starts to get earlier and earlier until the present time, and the average last fruiting date gets later and later. So in about 1950s, we had a fruiting season in southern Britain of about 30 days. But now it's, about 75 or more days. That's not only telling us something about fruiting patterns, but it's telling us about the behavior of fungi. This data set also showed us that mycorrhizal fungi behave rather differently to wood decay fungi. It showed us also that some fungi now fruit in spring that never did before. They've always been spring fruiters, but some now fruit in spring that didn't before. And this is particularly true of wood decayers and some of the litter decayers. We know that patterns are changing across Europe too. We know that the mean fruiting days for a whole range of fungi are changing. These plots are just for the sulfata fungus hypholoma. We know that distribution patterns are changing, and even there are changes in the hosts of fungi. Of course, pollution is important too. In the 1980s, it was sulfur dioxide pollution that we were all worried about. This is a picture of dead forests from the Czech Republic. Now, one of the major issues is nitrogen deposition. And nitrogen deposition, and indeed fertilizer addition, affects considerably the communities of mycorrhizal fungi. So there's a tendency for mycorrhizal fungi, which produce these extensive networks in soil, to be lost, whereas those which don't grow out to anywhere near the extent are sometimes retained. And this could be having dramatic effects on our trees. And then, of course, there is also loss of hosts for fungi, movement of biota. We are terrible as humans, moving plants and animals around the globe, spreading diseases and goodness knows what else. And then there are invasive species. And Anne Pringle is going to tell us about invasive species very soon. Of course, it's important because of these changes, or anyway, that we protect fungi. They are so important to us. We realise how important they are for trees, for wood decay, etc., etc. We can produce red lists of fungi. indicating those which are threatened, which have the greatest threat. We can try to conserve species of fungi or the habitat that these fungi occupy. There are wood decay fungi that are threatened. There are mycorrhizal fungi that are threatened. There are parasites which are threatened, and tree habitat is threatened too. There are a few examples here. For example, the Hericium species and Buglossopora important as wood decay fungi, or wood decaying wood is their habitat. And there are also mycorrhizal fungi threatened. Here are 3 examples here of stipitate hydnoid fungi. And trees themselves are crucial to our environment. Ancient trees and veteran trees have many, many habitat features illustrated here on this lovely diagram produced by Neville Fay, including fungi as habitat, but also rot holes of one type or another, sap flow, broken branches, etc., etc. Habitat, large areas of habitat are being threatened. Illustrated here is Atlantic woodland with the rare hazel gloves fungus. It's essential that our management employs methods for habitat conservation, for tree conservation, for fungus conservation, and Jenni Nordén will tell us about this. So we have 2 talks this evening. The first one is from Anne Pringle from the University of Wisconsin-Madison. Anne is an eminent mycologist who works on all sorts of aspects of fungal ecology and fungal evolution, but she's going to tell us this evening about the problem of invasive mycorrhizal fungi. Then the second talk is by Jenni Nordén from NINA, the Norwegian Institute for Nature Research. And Jenni has done a huge amount of research into wood decay fungi and to fungus conservation. And she'll tell us about the management for conservation of threatened wood decay fungi. So over now to Anne.
Thank you very much, Lynne. Lynne from the past. We'll be hearing from Lynne from the present later on. Of course, at the moment, that's Lynne from the future, but you know what I mean. Anne, let's just go over to you and get back to some proper, sensible discussion.
I'm speaking to you today from Wisconsin in the United States, and I want to start by acknowledging that our university occupies ancestral Ho-Chunk land, a place that their nation has called Dejop since time immemorial. In an 1832 treaty, the Ho-Chunk were forced to cede this land, and that history of colonization informs our shared future of collaboration and innovation. Today, we respect the inherent sovereignty of the Ho-Chunk Nation, along with the 11 other First Nations of Wisconsin. So I want to talk with you today about the problem of invasive mycorrhizal fungi. And I have a lot to say, and I'm going to be mindful of the time. This is something I've been working on for quite a long time. And even though I feel in some ways I've gotten a lot done, my lab has gotten a lot done, there's so much left to do. And it would be fantastic to have other people involved with thinking about these kinds of issues. So I want to start by pointing out something that may be second nature to a lot of people in the audience, or maybe not. And that's this idea that of the big plant, little plant experiment. This is a kind of experiment that's been done probably thousands or tens of thousands of times by now. And the idea is that you grow a plant with and without mycorrhizal fungi. You grow a plant species, individuals with and without mycorrhizal fungi, and with different species of mycorrhizal fungi. And these big plant, little plant experiments are the basis for what we think we know about mycorrhizal fungi. And over and over and over again, with lots of variation, lots of diversity in the responses, but in general, the main theme is that plants, and here you're looking at a Plantago species growing with a fungus that used to be named Chlorostroma Westlandicum, and these are 2 individuals that are growing with the fungus, and here are 2 control individuals, and there are lots of differences in how they grow. But at its heart, the message is that plants with fungi grow better. And I find that this message, which comes from these big plant, little plant experiments, has really permeated the world. So this isn't just something that academics talk about with each other. This is something when the arborist comes to my home and trims the trees, he's heard about mycorrhizal fungi. He's heard that they're good and that we should have them. So this is a movies like Fantastic Fungi. This is a message that we hold. And a lot of people know these things. And of course, I just showed you an experiment with Plantago, but it's also true that you can do a big plant, little plant experiment with trees. So here's the big plant, little plant experiment with oaks. And these are oaks growing with truffle fungi, and they're lovely and healthy, or without fungi, and they're not lovely and they're not healthy. So here's I'm going to even suggest that it's a paradigm. Plants with fungi grow better is the paradigm. I also want to just set the stage by discussing, and again, just maybe a bit of review for some people, but also just setting us on the same page in case it isn't review for some people, that there are really two distinct kinds of mycorrhizal fungi. So when you say mycorrhizal fungi, you're talking about fungi that are associated with plants, and very specifically with plant roots. But within that word mycorrhizal, there are really two distinct flavors, if you will. One flavor is the arbuscular mycorrhizal fungi, and these are fungi in the UK that would associate with grasses or ornamentals or your tomato plants. And these are endo because they're inside roots. So if you could peer inside a root, you would see structures that look like this, and you know you're working with AMF. But trees, and temperate trees in particular, we're not going to talk too much about tropical trees, a little bit. Temperate trees, so the trees you would have in the UK, associate with ectomycorrhizal fungi. And these are ecto, or outside the root. These fungi grow outside the root, and they form these lovely structures called root tips, which you can see with your eye. If you dig up soil and you look for roots, you'll see root tips, you'll see these fungi. So these really two very different kinds of mycorrhizal fungi. Now I want to tell you a story. And this story, we're going to go all over the world, but this story starts in the southern hemisphere. And in particular, we're looking at a photograph that he took out a car window as I was driving around in New Zealand. And in case you didn't know, pines are not native to the southern hemisphere. So pines, for example, are not native to New Zealand, but they're planted there. And here you see, if that looks like some protuberance sticking out of a hillside, it does to me. It doesn't look at all natural in that square with those, you can see there are rows of trees. This is a plantation, is what it's called, right? It's a plantation of trees, pine trees planted in this, plot of land in New Zealand. And so then you could ask, why are pines being planted? And this isn't just happening in New Zealand. It happens in South Africa. It happens in South America. It happens all over the Southern Hemisphere. And of course, the answer is economics. We need the things that trees can give us. So we cut down the trees and we turn them into paper and paper products. And this is a really enormous industry. And it, we could talk about that for a long time. So a backstory of these forestry plantations involves fungi. And it turns out when people first started planting pines in places like New Zealand or South Africa, they didn't grow very well. And they hadn't done the big plant, little plant experiments yet. No one had done that yet. And so people didn't totally understand at first what was going on, but eventually foresters figured out that if you added some soil from a place where pines were already growing well, somehow if you added the soil, that enabled pines to grow. So what happened is that soils were moved across the globe to enable forestry, to enable pine plantations. And like pines, ECM are not native to the Southern Hemisphere, and so you needed to move soils from places like England. I put this full citation here just in case you're interested in it. But this guy, Mikola, actually talked to a lot of foresters in places like South Africa, and he recorded what is essentially an oral history. And he would say, where did you get your soil? And someone would say, in this case, well, I had a friend in Oxford and this place called Blenheim Woods, which someone must know about in the audience. I've never been there, I don't know, but apparently it's a woods outside of Oxford. And someone shipped a tin of soil over to the continent of Africa, and that started off the pine plantations here. And et cetera. And so if you read McCullough, he documented, well, soil came from here, but then we sent it from one country to another. And then when it reached particular countries, it got sent all across, within the country from nursery to nursery. So these soils were moved to inoculate trees. And what that means is that, with the soil, ectomycorrhizal fungi were being moved, because that was the whole point. That was why they needed the soil. They needed the fungi. They needed the fungi in order to enable the pines to grow, in order to grow them for paper and paper products. So actually quite a while ago now, I became interested in this idea of this aspect of biodiversity that's being moved around the planet, and yet somehow we don't know about it. We talk a lot about invasive plants and about invasive animals and about disease, but we don't talk too much about mycorrhizal fungi being invasive. Somehow that hasn't permeated the consciousness of a lot of us, and we just don't think about that kind of biodiversity being moved around, but it is. And so here are a lot of different genera of fungi. And these are reports from the literature that I could find with my collaborator, Elza Blinga and Ben Wolf. And we could find, for example, the genus Amanita. It's a really charismatic genus of fungus, and it's been reported as introduced quite a lot, more than 80 times in the literature. I can find reports of someone saying, I'm finding this Amanita in this place. It has never been here before. And so when you find that, you can tick that as an introduction. And this black is the number of species that are involved in this total number of introductions. So the biodiversity is this chunk of biodiversity is being moved around. And we published this result quite a long time ago now, but I want to make the point that there's a database associated with this paper that you can download. So if you are watching this and you're in Australia and you want a list of the species that have been introduced to Australia, there's at least a start to that list associated with this publication. Here you're looking at a map of the world, and you're looking to see that these circles are proportionate to the number of introductions that have been recorded in the literature so far. For Brazil, for example. And you'll see that there's a lot of introductions associated with the Southern Hemisphere because of the plantations that were planted there, but not exclusively. There are other ways that we've moved fungi around. It's not just about moving soils to enable pine forestry, although that's certainly, that's the story I started with because it's an important element. So these things have been moved around. These species have been moved around. Not surprisingly, then, some of these species have become invasive. And so here you're looking at a poster, part of a poster that's put up around California, because in California, in particular around San Francisco, this species, Amanita phalloides, or the death cap, is rampantly invasive. When you walk in the habitats around Point Reyes National Seashore, there are hundreds and hundreds of these mushrooms. And by the way, they're twice as large in California as they are in the UK, which Lynne is nodding her head because she's seen them. And it's pretty gross and disgusting. It's a big garden of death that you're walking through. And this is a species that's spreading through the forests. It's associating with local California coast live oak. So it's not associating with, it's on its own. It's making its way by, on its own, associating with endemic plants. So it's a thing, and invasions are a thing. That's not the only place invasions have happened. And so I've talked about the death cap a lot through the years, and I want to point out that there are other stories. It's not the only story. So now we're going to shift to an entirely other country, Colombia in South America, where I've been working in collaboration with Natalia Vargas. You'll see her photo later. Now we're high in the Andes. So Colombia is a fascinating country for lots of reasons. Also, it happens to be the southernmost distribution of oaks in the world. So there's an endemic oak there, Quercus humboldtii. And you can see some of those forests of Quercus humboldtii here. But it's also a place where a Mexican pine, Pinus patula, has been planted extensively for forestry. And there's a history behind that, and it has to do with preserving the Quercus humboldtii forest by planting pines instead. But when you talk to foresters there, which we've done, they consider it essential when they plant pines to add fungi to the soil. And so they've done various things. And one of the things they've done is take sporocarps of the European Ammonite Muscaria, and you can talk to them. They literally take sporocarps and they mash them up and they distribute them on the pines as they plant them. So recently we've done a lot of really careful work to try to understand what's going on and to document that like the death cap in California, Ammonite Muscaria in Colombia is invasive. So now you're looking at a map of South America, so you can situate yourself as far as where Colombia is. Then you're looking at Colombia. These dark patches here are the two great mountain ranges, the Andes in Colombia. Green marks a plantation that where Natalia found Ammonite Muscaria. And blue is particularly interesting because that's one of the sites where we found Muscaria now associating with the native Quercus humboldtii, this very southernmost distribution of oaks. And it's rather amazing to go there. You're very, very high up in this mountain chain with these oaks, and then you see this European species. This is a photograph I took just at this site that you see in blue. So this is happening. Invasions are happening, invasive species. We have these kinds of data, which are somewhat the holy grail of invasion biology. You're looking at here, each dot marks a mushroom. So here you're looking at mushrooms we collected in a plantation of Pinus patula. Sorry, I'm having trouble managing my Zoom. Sorry. Here's the purple is Muscaria mushrooms with Pinus patula. Here it's a mixed plantation. And here is an invasion front. These are native oaks now with the European Muscaria. So it's rather incredible to have this kind of an image where you can actually see that the fungus has dispersed and made the leap associating from planted not native species of trees to a native species of tree. So we published this, and I won't say any more about it other than this photograph. This is not photoshopped. When I show this photograph, it looks like someone made little pictures of Muscaria and popped them in. I think that the Muscaria here, like the death cap in California, is very much larger than the Muscaria you would find elsewhere. I don't have the data to prove it. But when you see these Muscaria, they can be as large as a dinner plate. I've never quite seen anything like it before. And here you are. This is the kind of habitat. So this is ground zero for the invasion. And here's some other really lovely data just to affirm or confirm that the more forests you have, so here are hectares of planted forest in different countries in South America. The more planted forest you have, the more introductions you have. So Brazil leads the way with both planted forests and numbers of fungal introductions. And Venezuela is at the other end of the spectrum, and here's Colombia somewhere in here. So there's just no doubt that part of the dynamic of planting trees in the Southern Hemisphere has been bringing fungi and introducing fungi, even though I would say until very recently, this wasn't a thing that people were talking about. And I'm somewhat concerned that it's still not a thing that people are talking about as much as they potentially could. You know, because, for example, well, we'll get to that in a second. There are other ways to do this, is what I'm going to try to say. So why do we care about invasive fungi? Just in case, just in case that's a question, let's talk about that. So we think that we have names, and I think Jenni's going to talk about this much more eloquently than I can, for something like 100,000 fungal species. But we think there are something between 1.5 and 10 million fungal species. In other words, most of the globe's fungal biodiversity is undescribed. We don't know it. We don't know what it is. We could talk about where it is and our suspicions about that. So we're in a situation where we really, really don't have a handle on what kind of biodiversity we're talking about. We don't have names for it. We're in a difficult spot there. But we know in general that biodiversity is threatened. In the U.S., if you try to understand, what's threatened, if you look at plants, which in the U.S. there are something like 18,400 described species, 15% of them are categorized as imperiled. For fungi, there are something like 38,000 described species. Remember, we know that that's wildly inaccurate, right? We know that however many described species we have, there are many, many more undescribed species. And yet, even of those described species, only a few are noted as imperiled. So you can come to one of two conclusions. Either fungi are less likely to be imperiled than plants, or what I think is more likely is that as we don't understand fungal biodiversity, we don't understand what the trouble is. There's just a great deal that we don't know. But of course, part of the threatening of biodiversity is invasive species. So here is an image of two park rangers in the US pulling out garlic mustard, which is a plant in the US that a lot of people care about. It's quite invasive. I spend time pulling out garlic mustard. Lots of people I know spend time pulling out garlic mustard. And we pull out garlic mustard because we know it does damage to local biodiversity. And so why should we care about an invasive death cap or an invasive muscaria? Because of the very real potential for it to do damage to local biodiversity. So if the first rule of intelligent tinkering is keep all the parts, we're not doing that very well. We have no idea what we're doing. We're tinkering away without really having a sense of what we're doing. So the only piece of evidence I can give you, so this is again a dramatic image of the death cap from this poster that's put up in parks around California to warn people that the death cap is there. The best piece of evidence I can give you that I think that there is trouble is this. If you go, so here are two forests in California. I'm sorry, I don't know what that blip is. These are two forests in California. And if you go and you collect over a season and you just collect the mushrooms, just the above ground biomass of the mushrooms, you'll find that most of that biomass is death cap. Back to this idea of gardens of death and you can't put your foot down without stepping on one. This is fairly remarkable data. So the idea that in this forest over a season, more than 80% of the biomass of mushrooms that you harvest is death cap suggests that probably there could be trouble with the other fungi that are there. And these are really unusual, beautiful coastal maritime habitats in California. So the theme is we don't know what we're doing and there's trouble and we're moving things around. Now I want to switch the conversation. So this is a story I've been telling you about history and it may seem far away, right? Who would ever think of moving soils around anymore? And yet there's another dynamic emerging ongoing right now. When I talk to people in Wisconsin, arborists or gardeners, a lot of them are really interested in having mycorrhizal fungi in their garden. And they go to places and they buy mycorrhizal fungi and they spread products like this in their garden because they want those fungi. And it gets back to this idea of the big plant, little plant experiment. We all know that mycorrhizal fungi are good and we want them. Sometimes these products are really interesting. Here's a product that we use in our greenhouse at the University of Wisconsin-Madison. And I want you to notice that it has mycorrhizal fungi in it. We don't know what they are. We don't know if they're ecto or endo. We don't know if we just don't know what's in there, but they're mycorrhizal fungi. But good to know there's also a biofungicide. So you're using soil that has both mycorrhizal fungi and something to kill the mycorrhizal fungi in the same product. So just to say, it's hardly even buyer. I don't even know what to say. Buyer beware. It's a fairly nutty situation in my view, and a lot of very strong claims being made. So in this context, then, my lab received funding from the National Geographic to try to explore, and we're not the only lab doing this, to try to explore this idea of mycorrhizal inoculants and whether they're necessary or whether they work. And one of my students once said to me, adding mycorrhizal fungi to your garden is like adding salt to the ocean. And I've always thought about that, and I think it is often quite true. You are adding salt to the ocean. Potentially. However, there's a need for some careful work. So we're doing this work in a lot of places, but I'm going to bring you back to Bogota, Colombia, for the rest of the talk and to tell you this story. And of course, start by saying that sustainable cities, green cities, have trees, have plants, and this is why we care. And this is why we're asking the question at all. Do street trees need additional fungi? Do trees in cities need fungi? Because urban trees are essential infrastructure. They provide parks and habitats for urban wildlife like birds. They provide a lot of pleasure to people. They actually do good things for your climate. They minimize the heat island effect of cities. So just to affirm, I think I'm probably preaching to the choir here, that we need trees in cities. And so the question that we're exploring in the context of this National Geographic grant is do street trees, I should have added, do street trees need additional fungi? Do they need these products? Do we need to add something? Do we need to add fungi to these soils? And then really the big idea that we've been pitching and we're testing, and it's very preliminary and very in progress, and I don't have a complete answer, but I have enough of an answer that I want to start a conversation, is whether we need products or whether we could use whole soil from a healthy nearby tree to help start a street tree that you're just planting. And this is equivalent, I don't know if people have heard about this, to a fecal transplant, where you can have people in the hospital who are very sick from what's called a C. diff infection. And instead of using antibiotics or all kinds of manipulations, you simply ask that person who's about to die to swallow some poop. Sorry to be so graphic, but you ask them to swallow somebody else's poop, and or you shove it up their butt. Again, sorry to be graphic, and that's called a fecal transplant, and it works. And those people who are about to die get up and walk out of the hospital. And if you haven't heard of that, it might be something you want to Google. It's rather interesting. So with that idea in mind, maybe a whole soil transplant is another idea. And here is this really suffering street tree. This is actually in Morogoro, Tanzania, which is another place I've done some work. And yeah, this street tree is not doing so well. All right. So the first question then was the first experiment that we did. Sorry, I'm managing Zoom in the background and it's not going very well. All right, so the first thing that we did in Bogota is try to compare the fungal colonization of urban trees to the fungal colonization of rural trees, because it seems reasonable that the first thing you should ask and answer is, do the urban trees need fungi? I think we all sort of assume they do because they look like they're so much in trouble often, but do they? It would be worthwhile to collect data on that. So we went all around Bogota, which is an enormous city, and it was really a fantastic adventure, digging up roots. And also then we went to various rural places outside of Bogota and did the same thing. We're working primarily with Quercus humboldtii, which is what you saw growing up high in the mountains. And here's, for example, one of the habitats that we're sampling, which is a park in a really nice neighborhood in Bogota. And you can see my student Nora and my student John and Natalia, who did the work with Muscaria up in the mountains. And anyway, we're measuring and sampling there. Now, before I get to the results, I want to point out, and I left this slide a little bit in Spanish, that the way that the city of Bogota thinks about their street trees, they think about them as either being in soft surfaces, in other words, in a park-like setting like this, or in hard surfaces. So, for example, in a box on a concrete sideway. And it turns out that that's really important, whether you're thinking about street trees in hard surfaces or soft surfaces. So here's an example of a soft surface, and here's an example of a hard surface. So a soft scaping versus a hard scaping. So I'll get right to the data. We're doing this work in other cities and with other species, but I'm just going to show you the oak from Bogota. And what you see here is just an overview of the results. And from this overview, you would say, well, the forest certainly has greater colonization. Forest trees certainly have greater colonization, more fungi on their roots than the urban trees. But it turns out it's a little bit more nuanced than that. It turns out that those hard surfaces, and this might not be the result that you would expect. It wasn't the result I was expecting. These hard surfaces, these little boxes surrounded by concrete, they have plenty of fungi. But trees in parks and medians and these soft surfaces don't have fungi. And I would put out there that the strong hypothesis explaining that result is that in these surfaces, the grass is fertilized. And when you add fertilizer, the fungi go away. But no one's fertilizing these trees in hard surfaces. So again, this interplay of fertilizer and fungi, right? Analogous to nitrogen deposition, honestly. All right, so that's the result from that. And then the next idea that we're testing is this somewhat crazy idea. You could call it crazy, that I'd be okay with that. That actually, instead of buying a product off the shelf, what you should do is take a little bit of native soil, plant it with your baby trees, and then put those trees out in the urban setting. So this is the flow that we're imagining. And I want to emphasize, okay, so here's the setup. So we're doing an experiment with oaks. I'm going to run 5 minutes over time. Can John or Sophia, can you tell me, is that okay? Okay, Lynn says that's okay. Okay, all right.
Of course it's okay. Keep going.
Thank you. All right, so here are our treatments. Control, nothing done to it. Native soil, so the whole soil transplant, equivalent to a fecal transplant. Just fertilizer, which would be the standard practice that the city of Bogota would use. They would just add fertilizer. Or native soil and fertilizer. We're doing this experiment with oaks and native pines. They're not pines, they're retrophyllum, but they call them pines, but they're retrophyllum. They have nothing to do with pine. And I'm just gonna focus on the oak data. I also want to emphasize that we're not moving soils long distances for this native soil and this native soil fertilizer treatment. So here you're looking at the nursery, La Florida. This is the nursery that the city of Bogota uses for all of its street trees. And we are taking, here's Natalia again, we are taking soil from one of these trees that you can see in the beyond, behind these potted palms. So we're taking a little bit of soil from there and just a little bit. And that's what we're using in our experiment. And I just also want to say that the Bogota Botanical Garden and the city of Bogota has embraced this experiment. They want the data, they want to learn, and it's been an amazing collaboration. We are doing our experiment on trees that they need to put out in the city, and they're going to put them out on the city. So this is as real an experiment as I've ever done, because these are not just experimental trees. These are trees that are going to go live in the city of Bogota, and we'll be able to track them for their whole lives. So it's just been an incredible experience. Okay. Well, what's the answer? Again, in progress, tentative. Please don't take this as the final word, although it's such an intriguing result. It's amazing. So it turns out that the best thing to do for your tree as you're growing it in a nursery in this experiment with these oaks in Bogota, Colombia, is add native soil and fertilizer. So these trees grow better than the trees that just have fertilizer, and these trees grow better than the trees that just have native soil, and they definitely grow better than the control. So here's some data. I realize it's really complex, but I didn't want to just leave you with a beautiful image. I wanted to show you some real data. So for example, if you look at height, these are days 0, 9, 97. We're almost to a year now. And so if you just focus on the year data, the purple is the plus soil transplant plus fertilizer. And these trees are doing much better than any of the other treatments. Why? This is an unexpected result. I didn't expect this. I think what's going on is that if you add fungi and fertilizer, and it's not a lot of fertilizer, this is not and it's just one dose of fertilizer at the beginning. This is not a wealthy experiment, let's put it this way. And when you prepare street trees, it's not a wealthy thing. I think you're adding nutrients and you're adding the fungi that enable the plant to find the nutrients. I don't know if that's true, but that's my hypothesis. So I think by adding both, you're making your fertilizer far more effective because you're adding the fungi that enable the plant to find the fertilizer. And by adding the fertilizer, you're giving the fungi something to find. I think that's what's going on. And as I said, we're going to follow these trees as they're planted out into the city and throughout their lifetimes. And they're not going to get anything else. This is what they get. These are real street trees, and this is, so this is what they're going to get as they go out. So that'll be really, really interesting to see. And again, just to emphasize that this mycorrhizal fungi, these fungi came from a whole soil transplant. They didn't come there. It's not a product. It's not anything like that. All right, so this is my last slide. Fungal introductions lead to invasions. The spread of mycorrhizal fungi, as I talked about at the beginning of this talk, is historic, but it's also ongoing, because we are spreading fungi as we purchase and add biofertilizers to the soil. And there may be viable alternatives, although again, I don't want to pretend to you that I've solved the problem today. Instead, I want to say, let's start the conversation. And I want to thank Natalia Vargas, who persisted despite the pandemic. Our plants were not waiting for the pandemic to be over, and it was our one shot to get this done, and she did it. So I just really want to honor her and her work, and thank my lab. And I'm sorry that I ran over, but that's my talk. Thank you so much.
Thank you, Anne. Do not be sorry in the slightest for running over. I think you could have carried on for far, far longer, and everyone would have been very, very happy indeed. You can look at the chat when we've all finished, but try not to let your head get too big, because people are very, 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 very interested in what you had to say. Right, from one fantastic presentation straight on to another. Jenni, I would like to hand over to you next.
Hello, everyone. Yes, thank you. And that was really, really exciting and interesting. And thank you, John and Lynn and Sophie and all the organizers of this wonderful symposium. I'm really happy to get to participate in this one. Okay. I would like to talk about the challenging balance between intensive forestry and conservation of wood decay fungi in the Nordic countries, where clear-cutting forestry is the dominant form of forestry. I apologize for having a strong Nordic and boreal scope, but I hope and believe that the concepts are for the most familiar and applicable also in the UK and elsewhere outside the boreal forest. Yeah, I will first talk briefly about why there are so many threatened wood decay fungi, and then I will go through a few important aspects to consider in management for conservation. And the third part is about the current conservation toolbox of management in the Nordic countries. And I will end this talk with some management recommendations. So first, why are many wood decay fungi threatened? We have over 1,000 wood-inhabiting basidiomycete species in Fennoscandia, by which I mean Norway, Sweden, and Finland. It's over 300 species of polypores, bracket fungi, and over 800 species of corticioids, which are wood-inhabiting basidiomycetes without pores or stipes, often with flat and thin fruit bodies. In the Nordic countries, we often refer to red-listed species instead of threatened species because the categories near threatened and data deficient are also of concern. So the red list categories include regionally extinct, critically endangered, vulnerable, near threatened, and data deficient. Data deficient implies that we think that the species could be critically endangered or of least concern or anything in between, but we're unsure because we have too little data. Last year, I and my colleague Tom Helikofton assessed 800 species of corticioid and polypore fungi, 700 of which we were able to evaluate for the red list. Half of the polypores and 1/3 of the corticioids were assigned in one of the red list categories. The general trend during the last decades has been slightly negative. We have at least not yet seen any general improvement of the situation of the red-listed species, despite the conservation efforts and biodiversity-oriented management that has been done. The reasons for the decline of red-listed species include relatively rapid reduction in the area of natural forest and the associated decline in deadwood. forest landscapes have become fragmented. In Norway, 1/5 of the country is productive forest land, and over 90% of this is intensively managed. 4% is protected, and 5% is different types of temporary or permanent set-asides. Deadwood volume in the managed forest is today approximately 10% of that in a natural forest. For most redless species, clear-cutting-based forestry causes an almost complete transformation of their habitat. The habitat may become completely unsuitable or of poorer quality, and suitable dead trees are few or absent. Additionally, intensive land use causes fragmentation and spatial isolation of the habitat. I suggest that the main mechanisms of decline are decreasing local population sizes, decreasing local colonization rates, and increasing local extinction rates, and probably also decreasing reproduction because of lower probability of mating. Some species are affected more than others, especially the ecologically specialized species that are declining because of intensive forestry. Let's then talk about some important aspects to consider in management for conservation. And firstly, sufficient amount of suitable deadwood. And firstly, what is suitable deadwood? Many redless species need large trees and naturally died trees. Many of them are specialized in a single tree species. In Fennoscandia, it's often spruce, pine, or aspen, sometimes elm or ash. Some of them fruit only on logs in advanced stages of decay, and some have a narrow range of decay stages where fruiting is possible. Several redless fungal species are dependent on a particular other fungal species that grow on the log before them. Large trees are important because they offer a variety of long-lasting microhabitats for often slow-growing specialist species. Large trees have also declined the most because of forestry, and they have a long formation time. How much of the suitable deadwood do the redless species then need? They need many dead trees for a viable local population with many enough individuals to persist. High density of suitable dead trees also increases the likelihood of patch colonization. This is especially true for fungi because they disperse passively with airborne spores. So it's a matter of chance where the spore lands, and if we have many suitable logs, the chances of the spores landing on at least one of them increases. In different studies, deadwood volume thresholds of 20 to 40 cubic meters per hectare have been reported. For instance, in the study by Penttila, no redless polypore species were found in sites with less than 20 cubic meters of deadwood per hectare. We also looked into the effect of volume of deadwood based on a Finnish data set that includes 500 sites where 100,000 dead trees were systematically surveyed for polypores and a set of cotyledonous species. We made over 40,000 observations of over 100 species. We then asked, based on advanced analysis and modeling, how many species of red-listed and non-red-listed species do we expect to find based on these data if we survey 200 logs in sites with low or high volumes of dead wood while keeping all the other variables in their average values. And gray here means low volume of dead wood, and green means high volume of dead wood. It turned out that the positive effect of deadwood volume was more pronounced for the red-listed species than for non-red-listed species. We would find 0.2 red-listed species per 200 logs when the volume of deadwood was low, as compared with 1.3 red-listed species when the volume of deadwood was high. Let's then talk about favorable habitat conditions and continuity in them. Most red-listed species need closed canopy conditions. The microclimate is clearly more moist in shady versus open canopy conditions, and the drying effect of the forest edge on the forest interior microclimate may extend tens of meters into the forest, and therefore small forest patches are drier than large patches. Some redless species, however, prefer open and dry forest conditions. This includes species that are adapted to fire dynamics. Large patches are better in sustaining viable populations because large patches can sustain larger local populations that have a smaller risk of disappearing. Naturalness benefits redless species because it means suitable conditions now and also in the past, that is continuity in the suitable conditions. This continuity is important especially for slow-growing species and for population development in the long term. High forest age is beneficial because it means time for the build-up of suitable conditions and time for the growth of the local populations and for colonizations of rare species, which are rare events. Let's look at the Finnish study again and now see how naturalness and local microclimate influence the number of species that we find. As a proxy for naturalness, we used the density of cut stumps, and as a proxy for local microclimate, we used the basal area of living trees. Redless species turned out to prefer high naturalness and shady forests, whereas the non-redless species showed only a modest response to naturalness and actually were more numerous in open than in shady forests. Let's then look at the effect of forest age on non-red-listed and red-listed species, making use of a dataset from Norway and Sweden. Here, 1 on the x-axis means no response, no effect, and 2 means doubling of species abundance for each additional 33 years of forest age. The non-red-listed species become a little less abundant as the forest age increases, but the red-listed species benefit strongly from increasing forest age. Then we should talk about the importance of the area and connectivity of the habitat. I won't address the total area of the habitat per se here, but when I talk about connectivity, habitat area is included in the definition. For wood decay fungi, good connectivity means short distances between suitable host trees in suitable environments in space and time. Connectivity may impact each of the stages of the fungal life cycle. It has a direct effect on how many airborne spores reach a forest patch, how many of them have remained viable during their journey, and what the chance of successful establishment by spores is. It has been estimated that roughly 1 from 1 million spores landing on a suitable dead tree manages to actually establish in it. Connectivity also influences the probability of meeting a suitable mating partner. If you're the only individual reaching a distant location, then you have null chances of meeting a partner and mating, which is for most wood decay basidiomycetes necessary for reproduction, formation of fruit bodies. Connectivity may also indirectly influence the number of spores each fruit body produces, the mycelia growth rate, and the reproductive rate, for instance, through potentially reduced fitness in isolated small populations with reduced genetic variation. Fungi produce very many spores, and the majority of them land very close to the fruit body, but some may fly far. Dispersal in fungi is insufficiently known, but many studies have found indications of dispersal limitation at different spatial scales, even at the scale of a forest patch. For instance, Norros, here with blue, showed that the density of airborne spores of a specialist wood decay fungus was as low 80 meters from a fruit body as in areas without any fruit bodies. Those spores that do manage to fly far lose their viability during dispersal if exposed to too much ultraviolet light and cold temperatures. We collected spores from fruit bodies, exposed them to UV light in the laboratory, and assessed the proportion of the spores that germinated with or without a treatment. We found out that those species that we have found to be dependent on good connectivity based on occurrence data also seem to have spores that tolerate UV light worse than the spores of other species. So spores of connectivity-dependent species, which are typically redless species, lose their viability, ability to germinate more easily than spores of other species during dispersal when exposed to UV light. Let's then look at the Finnish data once again. Now we're interested in the area and distances of old forest in the landscape within approximately 5 kilometers from the local forest patch. This is S2. S3 is a historical land use gradient from southwestern Finland, where land use history is long and intensive, to eastern Finland, where intensive land use is relatively recent. The redless species benefit strongly from both of these types of connectivity, while the least concerned species show an opposite response or are indifferent. We see the same responses in our Norwegian-Swedish data. The least concerned species are on average indifferent with regards to connectivity to old forests, while the redless species are clearly more abundant in good connectivity conditions. What's more to this is that connectivity or high volume of dead wood are not sufficient alone. We need both. Here we divided the Norwegian-Swedish sites into 3 groups based on how large volumes of spruce logs they had. So these were all near natural forests and all had at least moderate volumes of spruce logs. When the volume of spruce logs is moderate, that is less than 29 cubic meters per hectare, increase in connectivity to old forest from left to right on the x-axis does not benefit the redless species. When the volume of spruce logs is intermediate, between 29 to 53 cubic meters per hectare, the redless species start clearly benefiting from improving connectivity to old forests. And when the volumes of spruce logs are very high, more than 53 cubic meters per hectare, the redless species benefit very much from improved connectivity. Note that the responses of the redless species and the least concerned species are almost opposite here, which suggests that competition might play a role here. Perhaps the least concerned species thrive in isolated forests because of less competition from the redless species that cannot reach the isolated patches. We can conclude that redless species are dependent on spatial connectivity and temporal connectivity or continuity because they are specialists, and the suitable trees for them are rare in space and time, which leads to resource limitation and which in turn strengthens the dispersal limitation. Redless species often have slow dynamics. It may take even decades from establishing to fruiting. Rare species have been shown to form fruit bodies more seldom than common species, which means low lifetime spore production and consequently low dispersal capacity. It's also so that rare specialist species need larger areas than common abundant generalist species to gain the critical population size needed for long-term survival. And it seems that spores of connectivity-dependent species may be less light resistant. Let's still look at the Finnish data once again to see how connectivity affects colonization and extinction rates in wood decay fungi. We went back to 1/3 of our Finnish sites approximately 10 years after the first survey and looked at what had happened. With this resurvey data, we were able to model the colonization extinction dynamics of 2 species. In many cases, there was no change in occupancy. The species was still present or still absent, but in other cases, the species had disappeared or colonized a site where it was absent in the first survey. Young forests acted as strong sinks here. They had many extinctions and very few colonizations, while the mature forests were weaker sinks or perhaps even sources. There were fewer local extinctions and more local colonizations than in the young forests. Colonizations depended in this case on stand age, connectivity, and number of dead trees. We then went back to Finland once again to resurvey more sites to get enough data for more species to be able to see how the level of specialization affects colonization and extinction rates. Here the species are ordered according to increase in specialization, and redless species are with red and orange shades. We found out that the redless species have clearly lower colonization rates than the least concerned species, and also somewhat higher extinction rates. The non-redless species are able to colonize also younger forests and forests with small volumes of dead wood, but the redless species colonize primarily old and dead wood-rich forests. This is especially the case if we look separately at the site where there is no known dispersal source in the vicinity, meaning the black curves here on the right that grow very slowly, indicating very low colonization rates, even with very high volumes of deadwood. Let's then look at the conservation toolbox of management, and firstly, the tools that are meant to help many redless species simultaneously. Protected areas is a traditional conservation tool, which usually means large and high quality areas. This sort of conservation is excellent, of course, but also expensive and often slow because there tends to be conflicts of interest. More recently, in the Nordic countries, voluntary conservation schemes have been established. They are quite popular, fast to establish, but tend to result in poorer quality protection areas than through state-initiated protection. And one can then question the cost efficiency and effectiveness of this type of conservation. Woodland key habitats and other small set-asides are spared from logging without compensation to the landowner. Therefore, they are defined as small patches. In Finland, they are typically only less than half a hectare in area, and in Sweden and Norway, somewhat larger. The small set-asides do host higher fungal species richness than ordinary managed forest, but they host few redless species, and it's also uncertain how well these small set-asides are able to maintain the small local populations of those few redless species that do occur in them. In the Nordic countries, it's common to lay living and dead retention trees on clear cuts. This is also a requirement of forest certification standards that cover more or less all managed forest land. Again, there's no economic compensation to the landowner for living trees, and therefore it tends to be the minimum requirements that are filled. It's usually small trees, mostly the tree species with least economic value, Fanascandia, often birch, that is spared. The retention trees are not monitored, and sometimes they disappear or get destroyed by harvesting machinery. Currently, less than 10 cubic meters per hectare of the dead retention trees are left, and it's few redless species that benefit from this practice because the logs are small, they're in open clear-cut stand, and there are too few of them. However, some redless species that are adapted to open and dry conditions may benefit from retention trees. Continuous cover forestry has the potential to produce large old trees in suitable microclimatic conditions that the redless species could make use of. Continuous cover forestry is, however, very small in Nordic countries. It's being discussed, but there is a strong resistance. Restoration activities such as deadwood creation, prescribed burning, and tree veteranization have the potential to help several redless species simultaneously, especially those needing burnt wood or slowly dying larger trees. For instance, Sclerophyllum carbonarium is dependent on charred wood. Then there are measures that are designed to help one particular species at a time. What are we supposed to do with highly threatened rare species that today only occur in a few or a single isolated old forest patch? Should we just hope for the best, that the protected areas will be established close by soon or enough, or should we accept that these species may not be able to wait for the slow protection processes and they join the galleries of extinct species? Well, we can actually give these fungi a helping hand so that we establish laboratory cultures, make mated mycelium grow into wood plugs, take the plugs into the forest, insert them into dead trees, and monitor the logs, and hopefully the species established, and this will in turn contribute to improved conservation status later on. This method has been shown to work pretty well, and it has the potential to be an effective way of helping the most threatened species. It's, however, important to keep in mind that translocation can only be an auxiliary of the primary conservation tool, which is well-connected networks of high-quality forest reserves. It's like first aid, which can be necessary in an emergency, but it's even more important to maintain good health, which is here well-connected networks of high-quality forest ecosystems. Translocations also involve risks. For instance, they may have unwanted impacts on the resident communities. Therefore, careful planning and monitoring is always needed. For a small number of fungal species, species action plans have been prepared. We did this in Norway for 13 endangered or critically endangered species, for which the majority of the European population resides in Norway. These are the species that Norway has a special responsibility for. Chlorostroma Westlandicum is one of these 13 species. It's an endangered ascomycete species, in Europe only found from Norway, where it was described, and recently also in Russia. The species is EN, also on the global fungal red list. It's a mycoparasitic species on the list is ascomycete fungus, Hypoxylon vogesiacum, and both of these fungal species are in turn dependent on elm as the host tree species, and the elm is also red listed. Chlorostroma Westlandicum has as threat factors the Dutch elm disease, deer, and forestry. We made a plan for actions that would improve the conservation status of the species so that it could be classified as VU instead of EN by the year 2035. This plan consists of protection of remaining unprotected habitat patches, limiting the access of deer to old elm stands by fencing or hunting permits, and counteracting the elm disease. The costs of this plan were assessed as high, but the authorities are anyhow planning to realize the plan, at least to some extent. Finally, some management recommendations. But before the actual recommendations, let's look at how the population of the same species that we looked at before are expected to develop the coming 100 years, assuming management as of today and assuming climate change. Again, the red-listed species are with red and orange shades. The red-listed species are expected to increase in set-asides, but decrease or remain low in managed forests. The predicted increase in set-asides is because of predicted increase in deadwood volumes in them due to climate change and improved tree growth. It's, however, unclear how the fungal species themselves will respond to warming climate. They may show a negative response that we were not able to include here. The current conservation measures in managed forests seem to be insufficient to help red-listed fungi outside the set-asides. To improve the situation of red-listed wood decay fungi, we must create well-connected networks of high-quality conservation areas and apply different methods of restoration to increase the suitable dead trees for red-listed species. Careful translocation may be justified for the most endangered species. In the forestry land, leaving larger trees and more of them would benefit red-listed species, especially in groups of living and dead retention trees. Increased rotation times in rotation forestry and continuous cover forestry in part of the stands would be beneficial. Larger woodland key habitats would be better than the current small ones. Everywhere, we should monitor the population trends of both red-listed and least concern species to get evidence of what measures work and to assess the effect of climate change, which is currently a big question mark in this equation. I end up with a close-up of a Kelo tree, an old pine tree. Many red-listed species are specialized in Kelo wood, and Kelo wood has a formation time of 500 to 1,000 years. Kelo trees are currently declining and perhaps vanishing for good with their associated red-listed species because of earlier management decisions. Thanks so much for listening.
Thank you very much, Jenni. Absolutely brilliant.
Thank you.
Look, see, the whole day was just building up to you three. That's all it was. They were your warm-up act. Don't say that. They might be watching. I'm not sure. Right. Okay. We've got lots of questions have come in. We're going to work through as many as we can. We'll kind of do what we did in previous sessions, which was I'll try and pick out questions for particular speakers. And then Lynn, you can sort of have a roaming brief chatting about whatever you like. Okay, well, there's loads of questions. Right, let's put a question in from Hang on a minute. I promise there are loads. I'm just getting confused. Okay, so Vicky has asked a question of Anne to begin with. Sorry, some of the questions are quite specific to when you were talking in the presentation, so it kind of feels a bit out of context later. But Vicky's asking, Anne, are the trees that you're using in your study native trees or non-native? And have you seen any differences between native and non-native in your study?
Good question. Also, can I get a copy of the chat later? There's so much going on there. I want to absorb it and I cannot. But there's some really interesting. Okay, good. There's some
we'll send a copy of the chat and all the questions to all of our speakers. Yeah, so you'll get hold of that.
Thank you. Okay, so I think that you're asking about the experiment in Colombia. And in Colombia, those are native trees. And it might have been a bit confusing because we're doing the experiment both with Quercus humboldtii, which is this oak that's native to Colombia, and something that they call Romaron pine, but it's not a pine. It's not a pine. It's a retrophyllum species, but they're native trees. So we haven't done it with non-native trees. And that's a really interesting question, whether you would find a different dynamic.
Thanks, Anne. Oh, Lynn, were you going to?
I was just wanted to follow up with the question while we were talking about that experiment, actually. That's the question I asked for you to ask. Anne, I was very surprised that it was the treatment with fertilizer and mycorrhizal inoculum. So I really want to know, what was the fertilizer? What was in the fertilizer?
Oh. I can get that to you exactly. I don't have it in my head. Actually, maybe if a question comes up for Jenni, I'll look at it. I have the exact detailed proportions. I can put it in the chat.
Okay. And so following on from that, I don't guess that you took samples for barcoding. It would be wonderful, wouldn't it, to know what the inoculum had in it when you put it onto the trees and what was there later on. For example, has that fertilizer really affected what mycorrhizal associates there were?
I agree. That would be really interesting. We have to be really careful. I mean, you saw what the pots are like. They're not very big. They're not even pots. They're plastic bags. And because these are street trees that are actually going to be planted in the streets, they're not, and it's a fantastic experiment that way, but it means we can't do too much we can't do any destructive harvesting because we don't want to damage the trees. They were very kind to let us do anything with them to begin with. They don't have enough street trees to meet the demand in Bogota. So unfortunately the answer is no, but it's something to think about for future. Yeah.
And also, just as a final thing before other people get more questions in, I would like to thank you for a wonderful talk, but both of you, but for emphasizing points that we've made in our discussions earlier in the day about not bringing in mycorrhizal inoculum from elsewhere, or indeed the potential for bringing in any invasive species from elsewhere.
Can I just add, I'm seeing in the questions some people who are kind of in the chat, some people who are kind of thinking about, oh, maybe I should try this, which I kind of feel like you should. I mean, we've been against using whole soil for anything because we've been petrified about disease. And clearly you have to use common sense about what kind of soil you're using and not bring it from too far away, et cetera. There are a lot of caveats. However, if someone in this audience tries this in the future, I would like to know about it, whether it works for you or not. Because this is one experiment that's in progress. That's a long way from a management recommendation and a long way from really understanding the dynamics. I just really want to emphasize that. But if people tried it, I'd be really excited to hear about it later.
I think in the Highlands of Scotland, they've done some replanting, probably of Caledonian pine. And I think that they use local inoculum in the same sort of way, and they've done various treatments. But I don't have first-hand information on that, but that'd be something worth following up.
Yeah.
Thanks, Anne. Thanks, Lynn. I feel like I'm not required here at all. Can I just switch my camera off and watch? That would work for me, and I'm sure it'd work for everybody else out there as well. Okay, question for Jenni from Paul. Paul is saying that plug spawn, as Paul's called it, plug spawn is becoming more popular, especially with introducing fungi to tree groups or woodlands. But the species that are readily available tend to be limited and usually edible. Is there any facility out there that could provide rarer fungi to be introduced by plug spawn? Paul says, I've only used Fistulina hepatica and Latioporus on oaks at present, but expanding the species range would bring the reintroduction practice to the masses.
Thank you for that question, and thank you everyone for such nice messages in the chat. It's lovely to read it. People are so kind. I don't actually know so much about what's available. In Norway, you cannot buy fungal inoculum from any store, but I know that in Finland there is a web store where you can order, for instance, Ganoderma lucidum and some common species and some a bit more rare species. But I actually don't know what's available. But I hope that everyone's really careful when when doing these inoculations, and reading the paper Lynne and I and others wrote last year about 10 principles for conservation translocations. There are important principles that are also important for other types of translocations of fungi. There are many risks that are important to consider so that you know what exactly you're translocating and what the effects of the translocation might be. For instance, how you might be influencing the genetic variation in the species, and whether it could potentially be a cryptic species, sister species of what you're thinking you're inoculating.
Thank you very much. Okay, a question for Anne from Tom. Hi, Tom in Belgium. Hope you're doing well. Tom asks, is the major issue with invasive mycorrhizae mainly associated with northern hemisphere species being introduced into southern hemisphere, or do you have similar data on a more local scale? And then Tom asks, what is probably a huge question, what is local for a fungus?
Yeah, that's a huge question and a really important one. So I'll start with that. What is local for a fungus? A really good example of our changing understanding of that is Schizophyllum, or the split gill fungus. Lynne, maybe there's a different name for that, but we used to have a sense that Schizophyllum was a species that you could find anywhere across the globe, and anywhere you went, whether it was Australia or the UK or the US, it was the same species. It was Schizophyllum commune. Now we know very well that that's a complex of species. And so the thing that we call Schizophyllum commune in Australia is not the same species, very likely not the same species as what you see in the UK. Another example, Amanita muscaria, the famous Amanita muscaria. You can collect something that's labeled Amanita muscaria in Florida. It is for sure not the same species as the muscaria that you will collect in Norway. No way is that the same species. But the problem is there are not enough mycologists to do the formal work of naming species, et cetera. So first of all, the idea of a truly global fungus, I think that's really rare, probably like Aspergillus, like some mold is, but mostly fungi are more endemic than the globe. That I feel comfortable saying. I think I also feel comfortable saying that there are some species that have very short dispersal distances that probably are local to something smaller than the size of Norway. But how small? I don't know. So I think that you're asking a question that's a really important one, and the subject of a lot might be stretching a lot of ongoing thought, I would say. I don't know about a lot of ongoing active research. But then the practical answer to your question is, for example, let's say you're planting a tree and you've decided you're no longer going to buy mycorrhizal inocula from the garden shop. Instead, you're going to try this whole soil transplant method. What soil should you reach for if you were going to try? And I would say the soil that's as close to where you're planting as possible, right? If find a healthy tree that's super nearby if you're going to try it. I'm not, and all the caveats that I've said before apply. We're far away from this being a management recommendation, but let's put it this way. If you were choosing between buying something off a shelf that's going to spread Glomus interradices everywhere versus using a little bit of soil from a block or 2 blocks away, I would vote for the use a little bit of soil from a block or 2 blocks away. Again, it's the start of a conversation. And now I forgot what the first part of the question was and whether I answered it or not.
The first part of the question, and I hate it when people ask this because I've always deleted the question just before, but it was, is the majority of the problem from Northern Hemisphere species being moved to the Southern Hemisphere?
I have yet again an unsatisfying answer, which is that we don't really know. This is the start, I would say, the start of thinking about mycorrhizal fungi as a thing that could be invasive was probably 15, 20 years ago. Before then, people really hadn't given it much thought at all. So I think we have these really few sort of well-studied case examples, the death cap in Muscaria. And then there's another dynamic called co-invasion. So for example, in South Africa, pines themselves are invading. And when they invade, the fungi invade with them. So it's called a co-invasion. And that's another dynamic that's being really well studied right now. But in general, I don't have like grand principles to give you, unfortunately.
Thanks, Anne. Okay, I'm going to ask a question directly at Lynne now, and then Anne and Jenni, if you've got anything, please chip in afterwards. But it's a good question that we haven't really covered today, Lynne, I don't think. Question from David, who asks, is there a relationship or interaction between the fungal communities and the ground flora, not just the tree species?
Well, yes, obviously. I mean, I don't know if you're thinking about just mycorrhizal fungi or if you're thinking about wood decay fungi, but obviously the ground flora have their own mycorrhizal associates. Very often they're arbuscular mycorrhizal associates, so they will be different. But whenever you've got fungi will interact with each other. So I mean, you've got a hugely complex scenario in and on the forest floor of the wood decay fungi, the litter decay fungi, any fungi that there are that can grow into soil. There's the arbuscular mycorrhizal fungi. There are the ectomycorrhizal fungi, and remember there are lots of different species attached to lots of different trees, and so all of those will be interacting. So there are obviously very complex relationships going on there. And if anybody like Anne, for example, wants to give a more specific answer than that, please feel free to. Or indeed, Jenni might have some comments on effect of ground flora on fungi in wood on the forest floor. I don't know.
I don't know about the effect of ground flora on wood fungi so much, but I wanted to mention that some wood inhabiting fungi grow in the soil and are mycorrhizal fungi, but then they climb onto dead tree to form fruit bodies. So that some species do that also.
And I expect we'll find more wood decay fungi that can be mycorrhizal and vice versa. I'm just thinking about the work in forgot what his name is now in Uppsala, where they screened at the wood decay culture collection, Jan Stenlid and forgotten his name beginning with F, and found that some of the wood decay species were able to form what appeared to be mycorrhizal associates. So I expect that the situation will be even more complicated than we already think it is.
Yeah, okay.
I don't think any of us want to think that this topic is more complicated than it looks. Thank you very much. Good question, good answers. Now, I was going to ask Jenni a couple of questions. Okay, a couple of questions, Jenni, about I guess some of the definitions and terminology that you've been using in your presentation. So I'm going to ask you two questions that I think are kind of related. One is, again, from Tom, asking what you consider to be closed canopy forestry and how large the cut areas can be to not impact too heavily on red-listed species. And then another question about definitions is from Danny, which is when you're talking about high age with respect to forest continuity, what are you thinking when you talk about age? Is that, you know, in years or decades or centuries? What are you regarding as high age when you say that?
Maybe I can answer the high age question first. In the Nordic countries, it's common to cut the forest when there are 60 to 120 years, depending on climate and altitude. But that's like middle-aged forest stand. It still grows rather rapidly, and they want to cut the forest when the growth rate starts to cease to maximize the amount of extracted timber. But for red-listed wood decay fungi, it would obviously be much better to let the forest stand for longer, so that there would be some dead wood that is being created. The first large trees start dying because of some disease or not really senescence before closer to 200 years, I guess. But spruces can live over 300 years easily, and pines can live up to 500 years, I think. So that would be a really old forest, from 200 years above. And then about the canopy gaps in the forest, I don't have so much expertise on that, because in the Nordic countries it's almost completely the clear cutting based forestry. So I don't have any number of meters in mind, but I guess everyone can imagine how a big opening in the forest may affect the microclimate. Half a hectare opening would definitely be quite large, I think. So in natural gap dynamics that takes place in natural forest, where individual trees die and maybe push over another tree when they fall down, I guess the gaps are, 10 meters or something like that in radius. And I think our old forest species are adapted to that kind of gap dynamics, and there would be both shady conditions and a bit more open conditions in the same forest if there are small gaps. So that would definitely be a suitable habitat for both old forest species and perhaps also for some species that tolerate the drier open conditions.
Fantastic. Thank you very much, Jenni. Okay. Lynne, did you want to say something? Sure. I'm watching you.
No, I'm positive. I'm very happy. Let's have some more questions.
Okay, Sam's got what sounds like quite a practical question for Anne, which is, could you expand a little on the soil's translocation process? How was it selected? How was it handled? How was it managed?
Yeah, and I'll add the caveat that I wasn't able to go for the day of the experiment. And so what I'm telling you has to do with what Natalia did with our collaborators from the Bogota Botanical Garden. And Lynn, I looked, but unfortunately I don't have the exact composition of the fertilizer. Unfortunately, I don't have that in my files that I can locate right now. So basically, I showed an image of a tree that's in a sort of remnant forest patch behind the nursery, and they more or less just scrape soil from the base of that tree. And we had looked at it before because the people in the nursery had never seen mycorrhizal fungi before, so we had actually done it was really fun. We went out and showed all the people who worked in the nursery a mycorrhizal root tip. But actually it was a leap of faith. We didn't do, other than using soil that was just near just near this nursery where the baby plants are, if that makes sense. We didn't do too much analysis. I mean, the whole point of this, in the way we thought about it originally, was that this would be something easy to do, where a nursery person, a forester, wouldn't have to do a lot of analysis. The idea is just to use local soil, and we didn't use too much, like per bag. It was mostly I can tell you that, at least it was nursery soil, 3%. So the control was nursery soil, 3% rice husk, and 5%, which is what I'm not sure why, and 5% sterilized native soil. That was the control. So in the native soil, it was nursery soil, 3% rice husk, and 5% native soil. So imagine a bag this big, and imagine 5% of that, and that's what we used. Again, I just want to caution that this is one experiment, and I really just want to say it's the start of the conversation. So I would really hesitate to say that's the way it should be done. It's the best way to do it. It's just, it's a little bit more than an idea at this point, but it's a little bit less than it's a lot less than a management practice. But yeah, that's what we did. I hope that answers your question.
And there's a question in the chat just at the moment which might be worth responding to. Someone's asking about how you know what mycorrhizal fungi are in soil. Would you like to answer that one, Anne?
Sure. Basically, think about it this way: we were using this native oak. I didn't say this. I should have said this. We were using this native oak as a street tree, right? That's the street tree. They're using the native oak. So we got soil from a native oak. And so in that way, again, we didn't do a detailed analysis of what was in there, but we know we're taking soil from around a Quercus Humboldtii and putting it with a Quercus Humboldtii seedling. And that would probably be the most effective way. You wouldn't want to go to a grassland and take soil from a grassland and put it with an oak, because grasslands are associated with arbuscular mycorrhizal fungi. But again, back to this idea of like the simplest thing is the best thing because people will actually remember it. The simplest thing to do is to get the most local soil you can from the same species or the same genus, at least. If you're a maple, find a maple, and a little bit of soil from that. That would be what I would try. And if you try it, let me know. Tell me what happens.
And to answer the question of how you would actually know which species there are, you'd sequence them nowadays. Oh. In the old days, you could look at root tips and you could look at the morphology and make a guesstimate of what's there. But nowadays we can sequence, so you wouldn't be looking with a microscope.
No, but sorry, I totally missed that. But most people don't have access to sequencing. We certainly didn't in this experiment. And so we just didn't worry about it. That's a really interesting comment in the chat from Martin Dixon.
You got us all reading the chat now.
I know, I'm sorry.
It's quite all right. Martin only addressed that to all panelists, so nobody else can see. Secret comment. While you're having a look at that, Martin, if you want to put that in the chat to everybody, then please do. Anne says, yes, please do. I've got a question for Jenni from Ben, which I think is quite an interesting question. Ben is asking, does the increasing number of fruiting body emergence mean that more fungal activity is happening? And if so, is that a good thing? If the fungi are doing so well, do they actually need protecting and conserving?
Thank you for that question. I'm a little bit uncertain if this refers to my presentation or is a more general question.
I think probably general about preserving and protecting endangered fungi, threatened fungi.
Yeah, well, most wood decay fungi are not doing so well, so it's definitely a great need to help them, protect them. But if a species produces a lot of fruit bodies, I think it's a sign that it's happy on that log. It's doing well on that log, and it has grown a large enough mycelium that enables the production. It has enough resources to produce fruit bodies, and it may be that it's both big enough and maybe the tree is becoming too decomposed, that it gives a signal to the fungus that now it's time to fruit and produce spores and leave the log because it's becoming too decomposed. But then it's totally up to how the rest of the forest is and how the landscape is. What happens if there's no other suitable log in the vicinity or within a suitable distance, then that's a dead end for that individual. And so we really need to make sure that there are enough of suitable dead trees at suitable distances of the known occurrences. So if the fungus is doing well on a single log, that doesn't tell us so much about the conservation need.
Absolutely, thank you, Jenni. Lynne?
And while we're on this, the idea of bigness and lots of fruit bodies being produced, I'd just like to go back to the beginning of Anne's talk, which is it's a totally different point from the threatened or endangered species point. You're right, Anne. When I visited California for a few months, I was absolutely amazed by the size of the fruit bodies, not just the ammonites. Everything was huge. The bolletes were this sort of size, and over here we always say that everything's bigger and better in North America. And when it comes to the fruit bodies, this absolutely was true. In fact, I thought the sky was bigger in North America, but it turns out I suddenly realized that everything was much more open, and I live in a little valley. So actually the sky is the same size, you'll be glad to know. But this business about fruit body size worried me for weeks and weeks. And I wondered why it was that the fruit bodies of Ectomycorrhizal fungi are so much bigger than we have over here in Europe. And so I don't know the answer, but I wondered whether it might be because, over here we have gone through our forests and replanted or managed or whatever. We've pillaged them for 2,000 years, basically, in continental Europe. And perhaps that's not the case in North America. Maybe we're often looking at old growth, or perhaps there's been one generation before, and maybe the soils are richer. Maybe the forests are more productive and can then feed fungi more sugars, and they can grow big. I don't know. I was just very much interested in your opinion on that.
I don't know. It would be a great undergraduate project to go to a herbarium, a really big one, maybe at Kew, and weigh sporocarps from all over the world and see whether there was some general trend. I mean, I know that about death caps, but because I haven't been in the UK that much. I don't know that about Europe, I guess is what I'm trying to say. So it'd be interesting to walk in Europe and see sporocarps and have that same experience of the opposite way. Oh, they're so little. Why are they all so little? I don't know. Yeah, I don't know. Remember one time Lynne came to visit me in Massachusetts, and we looked for networks in soil. And remember we had such a rough time finding them, and we realized that that the forests in Massachusetts are really different from the forests in Wales in terms of how you find networks. So I don't know. It would be great to have someone spend a lot of time in both places and think about that. Mm-hmm. I take your point about the management. There are still places, well, in Wisconsin, for example, in the summer or in the fall, really, I go up to a place that's never been logged before, ever. And I have to say that walking through a forest that's never, ever been touched that way, I had never, and this is acres, this is hundreds and hundreds and hundreds of hectares of land, and it's a really primeval experience. I've experienced that in the tropics before, but never in a temperate climate. And that was eye-opening to me. Yeah,
absolutely. And so yes, I mean, I think you're absolutely right. And when in Europe, I'm afraid we really don't have primeval forest. I mean, some places will lay claim to little bit small remnants, and Biologia we've mentioned earlier. But then again, in all the parts that I've been in, which isn't all of it, of course, have been managed. There have been some small bits in the Czech Republic I've been into, and it's a totally different from anything that we experience in any of our managed forests. That it takes you half an hour to walk 100 yards because you're clambering over these huge, great fallen trunks. So is it the same as that in North America?
No, I didn't see that quite the same way. That was very interesting. So there's a concept in conservation called shifting baselines, which perhaps people have heard of, or perhaps they haven't. And the idea is that your baseline is what you grew up with. That's what the world looks like to you. And then it can undergo radical change in your lifetime. Like my family's from North Carolina, and the North Carolina place where we go is completely different now from what it was when I was a kid. But my teenagers think it's great. They don't see all the dramatic development that's crazy and horrible. Their baseline is different from mine. And that's really important to fungal conservation, because sometimes people talk at me to say, I'm collecting baseline data. And I would say, really? Are you collecting baseline data, or are you collecting your own baseline data for the year 2020? Let's not pretend that whatever you're doing in 2020 is the same as what someone would have seen in 1800. That's a concept that I think it lives in the conservation world, but is perhaps less common in mycology, but it's really important. We're not collecting, fungi are not untouched. We're not collecting that kind of baseline data. Yeah.
Thank you all. Right, I'm gonna go against type here, and I'm gonna tap into a question that's gone into the chat. Normally it's a no-no, but I like this, so I'm going to put this to Lynne. A question from Federico, or a comment really. From an evolutionary point of view, can we consider Mycorrhizae the most evolved species of fungus?
I saw that one there and I thought straight away I shall pass that straight over to Anne. Anne, over to you. You're the evolutionary biologist.
All right, so here. Okay, what does it mean to be most evolved? I think that's the really difficult thing, right? I suspect that most evolved, and please put it in the chat if I'm wrong, has something to do with the most complicated or the most progressive or the most complicated. And in that sense, I would honestly say, who are we to judge complication? We used to think that this mycorrhizal symbiosis was very complicated. And it just seemed so, when people first started learning about it, it seemed so marvelous and so intricate. It must be something really rare and special. But guess what? The Mycorrhizal Association has evolved dozens or 80 or hundreds, depending on who you ask, of time. So there's been an independent evolution of that habit between fungi and plants many, many times over. So the symbiosis is not complicated at all. Biggest, most complicated genome, that's a different question. Fungi in general don't have complicated genomes as compared to plants, for example, which have really, really complicated genomes. I'm going to say that genome dynamics are often not about a species and the species interaction with other species or the environment. For example, it's often about what selfish elements a genome has within it and whether a species has evolved good defenses or not. So if you have a runaway dynamic of transposable elements proliferating through your genome, then you get an enormous genome. But it's not, you might call that complicated, but it's certainly, and it's most evolved for the transposable elements, I suppose, it's a good habitat for transposable elements. Sorry, I can't do this justice in a very short amount of time, but I'm giving it my all. So I don't think genome size is a good estimate, I don't think genome size, or I don't think number of different things is necessarily more complicated. Rust and smuts are really interesting because they go through so many stages, right? They have multiple different hosts, multiple different bodies and body types, if you will. So yeah, their genomes do a lot of different kinds of work. So if you measured most evolved by number of stages in the life cycle, if that were your definition of most evolved, it all hinges on what your definition of most evolved is. If that's your definition of most evolved, then rust and smuts would definitely be right up there, in my opinion.
Thank you very much. And if you need more time to explain it, you're all more than welcome to come back for another webinar. There's plenty of time. That'd be great. Okay, we've got about 10 minutes left, and I want Lynne to have a bit of time to give a little summary at the end as well. So let's just go for a couple of questions. I was going to go for the last question, but then Jackie, you've asked a question that you say is a simple question. Clearly not going to be a simple question, is it? But let's go with it anyway. I'm going to ask all three of you for your thoughts here. Jackie asks, could we survive without fungi?
That's an easy one. No. So, I mean, it's my stock answer to this is really fungi are essential to life on this planet for two main reasons. One is because of the mycorrhizal associations. 90% of plant species have mycorrhizal associations, which feed plants water, mineral nutrients, protect them to some extent from soil pathogens, and probably all sorts of other things. And the second reason is that fungi are the great decomposers of our planet, the garbage recyclers, if you like. And again, without them, we would be in a bad state because trees, as you know, have a lot of lignin in them. That's their main structural component. And there are very few organisms which can actually break down lignin. It's really largely a narrow group of basidiomycetes with some ascomycetes. We know some bacteria can, but they just do it on such a long time scale. It's not very effective. And so you need to be able to break down all that dead stuff, because otherwise the nutrients would be completely locked up in dead stuff and would not be available for plants. So for those two reasons, fungi are absolutely crucial to the functioning of terrestrial ecosystems and leading on from that ultimately to our own existence.
Thanks, Lynn. Anne or Jenni?
That was very well said, Lynn. I fully agree, of course.
I put in the chat a link to a National Geographic article that asks the same, that basically says fungi are key to our survival, and it talks about conservation. And I'm sorry, I think you have to enter your email in order to get access to the article, but yeah, it answers the same question. I think fungi would do very well without humans. I think if we disappeared, 99.999% of the fungi on the planet could care less. But I think if fungi disappeared, our world would fall apart in catastrophic ways that we probably have a hard time suspecting. And I think it's a great thought experiment. Plus, if we didn't have fungi, we wouldn't have the breads that we have, and we wouldn't have beer, just saying. So.
Wow, case closed. Brilliant, thank you very much, but clearly not appreciated enough. That seems to be the message. They're not as appreciated as other things. Right, there's just one more question I want to ask you, and I'm going to ask all three of you, then we'll go over to Lynn to close. A question from Daniel in Israel. Hi, Daniel. He's saying that Daniel has a good friend coming from a Mediterranean country in which pruning paints are still very common, as is spraying fungicide on fruiting bodies indiscriminately without even identifying them first. The common excuse of the old school arborists doing this is that their climate is completely different to the places that do the research, like Europe. Daniel's asking, if you had a couple of minutes in front of those people who just spray fruiting bodies indiscriminately with fungicide, what is the most important message you would deliver to them to influence them and change what they're doing? Lynn, I'm going to ask you first.
Right, so I would want to say don't, because you do not understand the roles of those fungi. We obviously don't know what fungi they are that they're spraying, but these things indiscriminately, they're going to be either decomposers or mycorrhizal fungi, probably. Okay, there may be a few pathogens in there, but who knows? And for the reasons that I've already articulated on why we wouldn't be here without fungi are the reasons why you should not be killing those fruit bodies. So fruit bodies, remember, are the reproductive structures, the sexual reproductive structures of the fungi, and they're the organs which produce spores and recombine genetic information to spread fungi far and wide. It's a very risky thing to do. You don't know what you're doing, I think. Anybody want to add further to that? I've forgotten the other bit of the question.
I would just say that the roles of fungi in ecosystems, that's a general principle true for any country, any place on Earth. The fact that fungi are decomposers and symbionts, that's general. You don't need research from a specific place to understand that. So given that that's true, you don't need a specific paper to tell you that you have to have fungi in your soils in order to have healthy plants. That's just a given.
And also, you wouldn't go out into the natural environment and batter down every plant that you saw on site. You just wouldn't do, you automatically wouldn't do it, or you wouldn't go out and pick every plant, every flower that you saw. And I think that you should think in the same way for fungi. Yeah,
or every insect. You wouldn't destroy every single insect that you came across because there are some insects that eat your roses,
Jenni, any thoughts on that?
Lynn and Anne put it very well. I don't have anything to add, but I missed the point. Why was this person spreading fungicide?
I think it's just the kind of concept of, in the same way as there's, some people when they make pruning cuts, they still just put the paint on it thinking it will stop the bad fungi getting in. And often those same people are just spraying chemicals all over fungal fruiting bodies to kill them because they're seen as a problem.
They're useful, they're helping us, they're important. Don't do that.
Well, that's a perfect summary, I think. Absolutely brilliant. We haven't got any more questions that we're going to ask, but Lynn, if it's not putting you on the spot, I was wondering if you could possibly just give us a couple of minutes of any closing points you wanted to make, reflecting on the whole day, really.
That was a shock, John. I hadn't thought of that one coming. Okay, so I think that really one sentence that I wrote at the very start of the book sums everything up as far as trees are concerned, and that is, a tree is never just a tree. A tree is always a tree plus very many fungi, and indeed other organisms too. And I think we've heard that throughout the day, both from the decomposers, the mycorrhizal associates, the endophytes, and everything else. Trees enhance all of our lives. We all know that, so that totally is preaching to the choir or to the converted. Fungi are as important as trees are, and I think we need to try and spread the word somewhat more for people to realise that fungi are important to trees, and that trees, as I said, are never just trees. And I'd like to thank everybody who's spoken today, all 10 speakers, which were chosen to reflect a specific aspect, I guess, of the book chapters, which go into more detail. I've thoroughly enjoyed the day, and I really would like to thank everybody. All speakers have been awesome, and this evening's speakers have been awesome too. And Anne has done a great job of talking about invasive species and pointing out that we really need to be far more careful as humans, as what we're doing, what we're moving around the globe. And equally well, if we'd had a plant pathologist talking about plant pathogens, I think they probably would have said the same sort of message in a slightly different way. And Jenni also has shown us how important it is to conserve fungi, and pointed to some of the ways in which we should be doing this. Some of the things which we've spoken about earlier today about large volumes of wood, etc., etc. I don't think I can sum up any more other than to say, just read the book.
Quite right too. Thank you, Lynn. Well, look, I guess, thank you, Jenni and Anne and Lynn, for this evening's webinar. I guess in closing, I'll just say it's been an amazing day. I hope you've all enjoyed it as much as we have. If you've got any thoughts or feedback or anything, then please do let us know. You can always email me, john@trees.org.uk. I'd like to add to what Lynn said, really, and thank you so much for joining us over the last 8 hours of webinars. And to our speakers, thank you to David and Christopher, Jill, Fred, Matt, Lisa, Rich, Dan, and of course Anne and Jenni as well. Thank you, all of you. And a very particular special thank you, of course, to Lynne Boddy, who was really responsible for all of this happening, who has written a masterpiece in this book, and it's brilliant for the Arbor Association to be involved in it. And Lynn, you don't need to hear this from me, but you genuinely are just one of the best science communicators I've ever seen, and you're inspirational. And honestly, it's just been such a privilege to work with you on this. So thank you so much. We're all very lucky. From this end, I'd like to say thank you to my colleagues who made today happen. So Sarah, Steve, Jack, Jason, Simon, and as ever, Sophie. And I should probably mention Ted Green as well. Thank you all for watching. It's been a great event to be a part of. We'll see you again in 3 weeks for the next Wednesday webinar. Please keep an eye out on social media and on our website for the Urban Tree World Cup 2021. Take care of yourselves, stay safe, and we will see you all soon. Thank you very much, and good night.